Good morning, everybody. I will start today's lecture. Today, we're going to discuss the stem cells in the reproduction system. Before I teach this class, this is an area I'm not familiar with. I didn't know that we even need the stem cells in the reproduction system because the capacity of reproduction is kind of limited, especially for females. So, but a lot of knowledge has been learned in this area, and so I'm going to focus today on two things. One's called the primordial germ cells, PGCs. The other is about spermatogenesis, how sperms are generated. Because in male, you have huge involvement of stem cells in the generation in the generation of the final reproduction cells. Well, in female, it is still a matter of debate whether adult stem cells are present there, or even if we have adult stem cells in the ovary. So that's still in the air. It's not decided yet. So we focus on the stem cells in embryo and the stem cells in the testes. So we start with an overview of early development, because the PGCs, the so-called primordial germ cells, arise really early. That's why I want to first have an overview of the early development. So at this point, you should be familiar with the terms. So the fertilized egg is called a zygote, and then the zygote will go through a process called cleavage. We call it cleavage because you don't have a change of the total volume of the embryo. At the beginning, the zygote is the embryo, but it's a single cell. So it's one egg fertilized. And then basically you're kind of cleave that big ball to smaller pieces. You don't have net gain of volume. It's the same volume for if we talk about the embryo itself. The volume is the same. So you just keep on cleave that ball to generate 2-cell, 4-cell, 8-cell until you reach morula. So the morula will be about 16 cells, 32 cells. In different species, that's different. So at that stage, we call it morula. So the morula is a compact ball of cells. So it's by cleavage of the zygote. And then you reach the stage of early blastocyst. And then the early blastocyst will take no time to become the mid blastocyst. At about mid blastocyst, you see a cavity there. The cavity is called the blastocyst cavity. The cells are squeezed to one corner of that structure. So you know that cell mass is called inner cell mass. If you isolate the inner cell mass, what you get is embryonic stem cell, the gold standard of all pluripotent stem cell. That's how you isolate embryonic stem cell. So here, another purpose I brought this up is for you to kind of appreciate the weakness of the definition of stem cell. So what really constitutes a stem cell? The pluripotent stem cell, the cells isolated from an ICM, apparently is stem cell by definition, because you can take the cell, take out the cell, put them into culture. Apparently, the cells will self-renew. Even if you have just one cell, such as in my laboratory, we culture the cell. Even if you just isolate one cell from an embryonic stem cell culture, they are going to replicate very, very rapidly. In no time, it will create a colony. So the self-renewal is of no question. And of course, if you give the right condition, they will become differentiated to give rise to all the cell types. So you fulfill the two criteria. One is self-renewal, the other is differentiation. So, but sometimes things start to become not as clear, such as the cells on the slide. Zygote, the cells of the two-cell stage, four-cell stage, eight-cell stage, up to the cells in the morula. Will you call those stem cells? Because probably there is no way, not as for now, you will be able to isolate those cells and put them in culture to show that they can self-renew. And of course, they can differentiate. Eventually, they are going to give an individual. Of course, they can develop. But to show self-renewal, it's not that easy for these cells. So their self-renewal ability is kind of transient, especially for zygote. I believe the zygote is different from the cells from the two-cell stage, but not by a lot. Not by a lot, because the process is called cleavage. You keep on, cleave the big ball to smaller compartment. That's all you do. So maybe the cells are not so different, but I believe the zygote and the two-cell stage cell will have some slight difference. Therefore, will you consider those, the cells in the two-cell stage, four-cell stage, eight-cell stage as a stem cell? Now, I would think in most the minds of most scientists working in this area, they will think these are stem cells. They're the only cells that you can call totipotent, because only these cells can give rise to an individual, including the extra embryonic tissues such as the placenta. So only these cells up to the stage of morula can do that. If you take the inner cell mass, they can't. They can only give rise to the embryo proper. That's the individual like us, but not the placenta. So therefore, ICM cells is not totipotent. They are pluripotent, not totipotent. But so if you say there is totipotent stem cell that can only be these cells, the cells from the zygote two-cell, four-cell, morula stage cells. So they are considered stem cells, but if you want to apply the strict criteria, well, maybe they're not. The definition is not strictly applied to this situation. So just some thing here for you to consider, because in the next slide, I'm going to talk about the primordial germ cells. They also exist in a transient manner. Therefore, you whether you call them stem cell, it is it's in a similar situation. So usually we still say them as a stem cell, but remember there may not be a perfect definition for stem cell. There is those rooms for you to wiggle. So what is primordial germ cell? So they are apparently the ultimate ancestor of all the germ cells by name. It's the primordial germ cells. They are kind of separated from other cells very, very early during development. The reason is understandable because the purpose of most species, the ultimate purpose of of most of the species will be to pass on the genes of themselves by reproduction, right? Even if they have some mutation, they have some changes in their genome compared to other individuals of the same species, that particular individual will like to pass on the genes. That's why you see the animals fight for mating because of that reason. That's we say a perpetual goal of life is to pass on the genes. Therefore, it's no wonder that at a very early stage during development, for example, in mouse, at around 3 to 5 days, about 3 to 5 days, the embryo have already set aside some cells to be the germ cells. For sexual maturity, it takes very long time, no matter for mouse or for human, it takes very long time. For humans, it's going to be for boys and girls, only when they reach their teenage years, they are sexually mature. But imagine at a very, very stage of development, the body have already set aside some cells to become germ cells at the very beginning of development. How early? That this will show to you. So basically, let's start with a fertilized egg as shown here. If you can see the arrow on my mouse. So that's a fertilized egg, and then that's fast forward to the stage of blastocyst. You have an inner cell mass. If you isolate the inner cell mass, you have embryonic stem cells. But just 2 days later, or 2.5 days later, the development will reach the stage of epiblast. Shown here, this cylinder-like structure is an epiblast, and the green part will be the embryo proper. That's the part that will eventually become an individual. While the top part, it's called extra embryonic. The white part eventually will become the placenta and other tissues that support, support the development of the embryo. Only the bottom part is the embryo. And then during this stage, epiblast, quickly the embryo will go through gastrulation. Gastrulation means now the embryo will form 3 germ layers: endoderm, ectoderm, and mesoderm. And at almost the same stage, or maybe even earlier than formation of 3 germ layers, some cells at the back would say that's the posterior region. So you observe this embryo at this stage is a gastrulate. During gastrulation, the left will be the anterior region, while the right will be the posterior region. So basically, at the back of the embryo, we call this the primitive streak along this line. So at the top of the primitive streak, there are about six cells already set aside. And these 6 cells are called primordial germ cells. So this will eventually migrate, because at this stage it is a single cluster of cells, but it will move bilaterally from the side. It's bilaterally because eventually the cells will migrate toward a structure called the genital ridges. We say the ridges because there are 2 genital ridges. Eventually, no matter it's for female or male, the reproduction cell will be in a pair of organs, like a pair of ovaries, a pair of testes, right? That's because the primordial cells will migrate bilaterally, symmetrically to the genital ridge, while the genital ridge will eventually give rise to the reproduction organs. Okay, so that's what's shown on this slide. In a gonad, that's when the migration has completed. It reached the genital ridge to become the gonad. And then eventually the gonad will further develop into ovary for females and then testes for males. So that's a brief overview of the formation of primordial germ cells and their migration. But here, one important thing is the marker of primordial germ cells. So the marker actually is OCT4. You probably will be surprised, because previously when we described OCT4 is one of the factors that Shen and Yamanaka used to reprogram cells, reprogram fibroblasts toward pluripotency. So OCT4 is associated with pluripotency. You also learned from me, from my lecture and student-led discussion, that the dose of OCT4 is important in stem cells, because the normal amount, which is the expression of OCT4 from both alleles, will kind of prime the cells toward the mesendoderm. So the one function of the stem cell is to differentiate. Therefore, they're always ready. So they self-renew, they keep their status, but they're always ready to jump out the door and then start to differentiate. That's why they keep OCT4 in the normal level, because in one of the student-led discussion, you learned that if you cut the OCT4 dose to half, now the cells are more pluripotent. I'm sorry, they have more ability in self-renewal, but they have a harder time to get out of the door and start to differentiate. So you learned that OCT4 is important, and the dose is important. But here I want to point to you that OCT4 is a marker of primordial germ cells. How so? And let's see this illustration again. Basically, this is an experiment done by putting GFP, which is an indicator, into the locus of OCT4. Remember that this strategy is not called linear tracking. So this is just a reporter of OCT4 gene, because you put that gene, GFP, the gene for GFP, into the locus of OCT4. But remember, OCT4 is a marker for stem cells. In most somatic cells, you don't have OCT4 expression. Or in other words, let's say in pluripotent stem cell, embryonic stem cell, or cells in the cell mass, you have expression of OCT4, therefore you should have green cells, right? Just like shown here. If you isolated the cell, the embryonic stem cell should be green because you have that genetic manipulation in place, so the cells should be green. But later on, even if the cell reached the epiblast stage, you still have the expression of OCT4 because epiblast is still considered to be pluripotent. Okay, so at that stage, you still keep the expression of OCT4. But later on, throughout the body of that embryo, you don't see the expression of OCT4 any longer because OCT4 is not expressed in differentiated cells, right? Only one exception. The exception is primordial germ cells. The primordial germ cells will keep on expressing OCT4, therefore the marker will be on in primordial germ cells. So during their migration into the gonad, and then later, once the primordial germ cells become the cells in the ovary, they will always express OCT4. Okay, so OCT4 is a marker of primordial germ cells. So you can track primordial germ cells by labeling OCT4 with a GFP marker. Okay, so now you have learned that GFP is not only a marker for pluripotent stem cell, but it is also a marker for primordial germ cells. And later on, once the primordial germ cells is in the ovary, they will further differentiate into the follicle cells. So that's what's happening in the ovary. In the testes, they will become the spermatogonia. Later on, we're going to touch about that. So here, I want you to learn that OCT4 is a marker. And remember that strategy shown there is not linear tracking. It's just a reporter for OCT4. I guess I emphasized quite a few times distinguishing the concept of linear tracking and reporter, and understand the different use of them in different contexts is extremely important. It's one of the key thing I emphasize for this class. So, the concept of primordial germ cells. At quite some time during embryogenesis, you have the identity of primordial germ cells. The primordial germ cells turn out to be pluripotent, just like embryonic stem cells. So they are considered to be one source of pluripotency. So on this slide, I listed 3 sources of pluripotent stem cell. If we are talking about animals. Okay, you have other ways to generate pluripotency, such as somatic cell nuclear transfer or reprogramming using Shinya-Yamanaka's factors, and later with other factors. You can do that in vitro, or That's an artificial way, right? You're manipulating a normal cell to make them become reprogrammed. But here, I listed to you 3 natural ways of getting pluripotent stem cell. So the emphasis, of course, is about the isolation of primordial germ cell. You know, basically at any stage, but most conveniently, you can do a E10.5 mouse embryo isolation, so you can get the primordial germ cells. You know where they are. You know, at the beginning, they're going to be at the back, at the top of the primitive streak, and later on they're going to migrate. So you know where they migrate, and especially when you have a marker such as OCT4 as your indicator, you can get those cells out and put them into culture. And those cells can develop into anything except the placenta. They're pluripotent, just like embryonic stem cells. They are almost identical to embryonic stem cells, just that the source of isolation are different. Okay, so the third way, of course, you can do the embryonic carcinoma cell isolation. So you have some kind of a tumor developed in the testis. That's called the embryonal carcinoma. And you can isolate the cell. You can put them into another mouse. It's going to generate another teratocarcinoma. You isolate them again. You can get pluripotent stem cells. So 3 ways here. The emphasis is about the concept of PGC. So the PGC is the ancestor, is the common ancestor of the cells in the reproduction organs, the most important cells in the reproduction organs responsible for reproduction. Their common ancestor is called primordial germ cells, and that cell is pluripotent. Now, I will move on to the discussion of spermatogenesis. But first of all, I want to point out, all the stem cells in the testis, that's the origin of spermatogenesis, was originally, of course, from the primordial germ cells. In the female organs, the oocyte is also from the primordial germ cells. But I fast forward, I didn't describe how the primordial germ cells will differentiate and eventually give rise to the stem cells in the testis. That is not the content we want to cover. So you must know that primordial germ cells give rise to the stem cells here. But first of all, I want you to appreciate the power, like we discussed in the hematopoietic system as well as in the small intestine. In those organs, the cell replacement rate is very high. It's about 10 to the power of 11 to 12 cells per day. That's a lot of cell replacement there. But here in the testis, that replacement rate is also very high. Although the whole process, the differentiation from the stem cell toward the mature sperm would take 70 days or so for humans. Okay, it takes 70 days to complete a single pass of differentiation, but it is extremely powerful. Like shown here, the yield is about 5.5 million cells, sperm cells, per gram of testis per day. The per gram of testis tissue is not one testis, just 1 gram of a testis tissue per day will give rise to 5.5 million sperms. But in contrast, the stem cells within the testis is a rare cell population. Here you can see the number is about 20 to 30,000 cells per adult mouse testis. That's really small, right? It's a small number, only 20 to 30,000 cells in one testis, but they're able to give rise to that many cells per day, per gram of testis tissue. So this is almost as powerful as in the other self-replacement systems we have covered. So here I want to give you a brief overview of the cells, the cell types in the testis. So we start with the structure, the macro structure. You see epididymis. That's where the sperms are collected. That's called epididymis. And then you have this structure. It's like a lot of tubules there. That's called a seminiferous tubules. The seminiferous tubule. Here we have a cross section of it. So basically, if you do a cross section, you see a tube-like structure, and in the center is going to be the opening, where things will flow, and then the rest is the structure as magnified here. So there are support cell called a Sertoli cell. That's the cell that are considered the niche cell there, right? They interact with the cells responsible for reproduction, right? So the stem cell here is also named spermatogonium. Spermatogonium. Later on, we're going to appreciate the different kinds of spermatogonium, but at this stage, you just need to know that they are the stem cells. They are close to the membrane, like shown here in B. They're very close to the membrane of that seminiferous tubule. They're close to the membrane, not close to the cavity. They're like very close to the membrane there. And then the cells will proliferate and differentiate, give rise to primary spermatocyte and then secondary spermatocyte. And eventually, the cells will go through additional differentiation and meiosis. At the beginning, it is just mitosis. The cell proliferation is by mitosis, but later on, at the final stage, they will go through 2 stages of meiosis and then give rise to the mature sperm. And in between sperm and the secondary spermatocyte, we call the structure there, or the cell there, spermatase. So that's an overview of how the cells will differentiate and migrate towards the tube and then release the sperm there. And next, I'm going to describe to you the hierarchy of the cells in this complex process. So one thing I want you to compare the here the name. Remember, the name is called spermatogonial stem cell. Spermatogonial stem cell, not a sperm stem cell. Spermatogonial stem cell. And as in other hierarchies, stem cell hierarchies, such as hematopoietic stem cell, and here the cells are also heterogeneous. Even they're all called stem cells. You probably will have the most immature stem cell, but when the most immature kind of differentiate a little bit and giving rise to other cell types, the other cell types are still considered a stem cell because those cells can still self-renew and differentiate. So you have different tiers of stem cells within the hierarchy. So this is probably common for a lot of adult stem cells, such as hematopoietic stem cell. When we discuss it at the beginning, Westman described the long-term HSC and short-term HSC, but those are all stem cells. We won't say that the most immature is stem cell while the others are not. For the same reason here, we have a heterogeneous stem cell population, quite a few heterogeneous stem cell population, even within the spermatogonia. The spermatogonia is the out layer. It is the out layer of that structure, close to the membrane, but still you have a lot of different cells there, not just one cell type. Of course, then one important question that scientists will try to answer is, what is the ultimate stem cell? Okay, you want to pinpoint to the tip of the hierarchy, the apex of the hierarchy, and then identify those cells, because for whatever conceptual or medical purposes, those are probably something you want to isolate, and those are probably most useful, okay? Because they are the ancestor of everything else. Okay, but you must understand the concept. In adult stem cells, often there is a hierarchy. So here we illustrate the different cell types within the spermatogonia. Those cells are indicated by either blue or orange here at the top of the illustration. Later on, you have spermatocytes, spermatids, and spermatozoa, and those are not considered stem cells any longer. And here, the cells can be grouped into three classes: type A, intermediate, and type B, spermatogonia. But apparently, the stem cells are limited to the tip of the hierarchy. So we call the stem cells asingle because they will exist as a single cell, isolated from others. And you have apaired, a pair of cells, they're close to each other, and then you have A-aligned. So from asingle to apaired to A-aligned, you found three populations of stem cells. So in terms of how immature they are, you will say that asingle is the most immature because it should give rise to apaired and A-aligned sequentially. But that's not the, that's a straightforward prediction there. But there are other theories. Like some others propose that the A-aligned will be able to give rise to asingle. So once you complete this cell proliferation and differentiation at the same time, the A-aligned can go through asymmetrical cell division, giving rise to asingle as well as A-aligned. Okay, that's some theory there. But to make it extremely simple, you can consider that asingle give rise to apaired and apaired give to A-aligned. So these three type A cells are considered to be the undifferentiated spermatogonia. So these cells are kind of identical. So they have the highest capability of self-renewal. And then next, there are 4 tiers of cells called A1 to A4 cells. They're still type A cells, but they're somewhat differentiated when you compare to the asingle, apaired, and A-aligned cells. So strictly speaking, from A1 to A4, they are no longer spermatogonial stem cells any longer. Okay, so just conceptually, they shouldn't be considered the stem cell population within the spermatogonia. But then from A1, A4 cells, you're going to have so-called intermediate spermatogonia and then type B spermatogonia. And eventually, you're going to have spermatocyte. And from spermatocyte, this is the point you stop to proliferate by mitosis. So previously, all the cell generation is by mitosis. At this point, it is going to go through 2 rounds of meiosis. Therefore, to make the spermatozoa, or in simple terms, sperm, to be haploid or 1N, because eventually this haploid will combine with the haploid from the oocyte, therefore creating 2N again. So here we have an illustration of the hierarchy. I believe this is the reason why the stem cell, although the number is so limited, right? The number we said it is extremely limited. It's for mouse per testis, you're going to have 20 to 30,000 cells, but eventually you can generate 5.5 million per gram per day. That is because of the proliferation involved. You have so many tiers. At each tier, you amplify the cell number at least. And here, we say that the AS cell, asingle cell, is the ultimate immature cell here. And if you put all the cells in the testis together, and then calculate the the ratio of this a single cell, it is only 0.02 or 0.03 percent of all the cells there. So it's a very, very rare cell population there. And here I want to point to you three scientists who made a great contribution in the study of not only spermatogenesis, not really just that, because these scientists did their work early, especially Charles Philippe Leblanc. That's the scientist shown at the far left. If you still recall, in the previous lecture, we introduced a theory of intestinal stem cells. So we have two theories. One's called we'll have two theories. The second one is called the stem cell zone model. Who proposed that model and eventually established that model? It's this scientist Leblanc. And he is a really important early figure in cell biology. I would say like a lot of the other work we described so far is based on the foundation that he has laid out. So he was one of the first to use radioisotope labeling of DNA. So he was able to label the DNA, therefore study cell turnover and the mitotic rate of cells. So because if you use, for example, like a 3H-thymidine to label the DNA, now you can study the rate of incorporation of the rate of DNA synthesis. So that's a good indicator of cell proliferation, because more incorporation will mean the cell will have to replicate their DNA very rapidly because the cells are dividing. If they're dormant, they're in quiescent, they don't have to take the 3H-thymidine. They just use the same chromatin throughout, if they just perform function without proliferating. So this scientist was one of the first to label the DNA with radioisotope. Therefore, he provided the foundation for our understanding of cell cycle, asymmetrical cell division, and of course, stem cells, because stem cells use asymmetrical cell division. They differentiate to give rise to progenitors while they keep their identity by generate another copy of themselves. Therefore, the division is asymmetrical. So he studied that. He basically laid out the concept of stem cells. So he and his student in 1953. Pay attention to that timeline. The 1953 is earlier than almost anything we have described so far. Okay, the hematopoietic stem cell, embryonic stem cell, even the somatic cell nuclear transfer all happened after 1960s. So he basically, laid the foundation of the concept of stem cell. And of course, he proposed that theory, the stem cell zone in small intestine. And I would say, later on, he was the one who proved the function of satellite cells, the muscle satellite cells. So it's crazy, one scientist can do that much in quite a different areas. Therefore, have a great influence to the scientists of later generation. So the Clement was the co-founder of that theory together with Leblanc. So the third scientist here, Brinster, was one who studied teratocarcinoma, who studied how to use embryonic stem cell as a tool for genetic engineering. So somehow he wasn't the one who won Nobel Prize, but he made great contributions in that area. He's one of the scientists who missed, but whose contribution is extremely important. And he was the first one to do spermatogonial stem cell transplantation. And he was able to show the single cell origin of sperms. Basically, he transplant from one animal to the other and then show the reproduction cells are derived from a single cell. I guess at this point, you start to appreciate the concept of the importance of a single cell, the single cell origin of stem cell, because that's a very powerful way for us to demonstrate both self-renewal and differentiation. Because usually only when you have a powerful self-renewal and differentiation, you can push one cell toward a colony, within organs, many cells within organs. It involves proliferation as well as differentiation. So if somebody tells you that they have identified a new stem cell, you ask them, can you demonstrate that a single cell can give rise to the intended somatic cell type in vivo, in an animal? That's the ultimate test. As we have already seen in multiple occasions, that's the task that the scientists consider to be a major milestone in defining adult stem cells. So here, I would like to tell you the identification of the ultimate SSCs. So although the scientists showed that there are stem cells in the testes, but to functionally demonstrate that wasn't a simple task. First of all, it's very hard to describe that hierarchy. It's very difficult to identify the cell. That's the first major obstacle there. And secondly, even if you identify the cell, you were able to capture the cell into your culture vessel, you can get them. And now the transplantation is another major task, which was overcome by the scientists that I showed on the previous slide. But imagine, most times you can inject the SSC, the SSC into the tube. That's let's go back to the structure. What you can do is inject the cell into the tubule, the seminiferous tubule. That's the only thing you can do, right? When you do a transplantation. Even if with some modern technique, it's still very hard to inject it into the membrane area without causing damages, right? So usually you put into the tubules, and then now you put into the tube, and now the cells, the stem cell you injected will have to travel back all the way to its niche for a successful transplantation, which is not easy because although it's soft, the tissue is not that solid, but there are a lot of tight junctions between the cell types here, especially within the Sertoli cell. So to go through the cavities of other cells, eventually move toward the final destination is not easy. It's very, very hard. That's why it takes a very long time to eventually demonstrate the function of SSC and also to identify the ultimate SSC. Okay, so but in early days, people know that the SSC should express KIT, the KIT. The KIT is also a marker for hematopoietic stem cell, as you learned previously. And scientists has known that KIT is a marker, but the KIT is a marker not for the immature spermatogonia, but rather when they move toward differentiating spermatogonia. That's A14. Okay, only when they move toward that more differentiated spermatogonia, they express higher c-KIT. Therefore, the KIT is not a good marker. Although it is a marker, it is not a marker for the ultimate SSC. And scientists also identified other defining markers such as alpha-6 integrin, GFR alpha-1, PRZF, and VASA. Especially VASA is very famous to describe the reproduction cells. But the later marker, this defining markers, integrins, these are either not extremely specific or they are not surface markers. Okay, they are not easily identifiable by antibodies. Okay, if you want your antibody to penetrate the cell, you have to kill the cell. So usually, if your antibody can detect a surface antigen, that's the most convenient one. And later on, sometimes scientists describe the presence of ID1. ID1 inhibitors of differentiation, if you call it all, but it's just a name. ID4 is a protein present on the surface of a single spermatogonia. So from that point on, now the scientists have a tool to demonstrate that. So this scientist, whose name is Oatley, described in not long ago, it's only 2017, that's about 4 years ago, he demonstrated for the very first time using a transgenic animal. Basically, he put GFP to the locus of ID4. Okay, so ID4 is a surface protein there. So he put this into the locus of ID4, and he was able to show that the AS spermatogonia with distinct levels of ID4. So ID4 bright, ID4 dim, and ID4 intermediate based on the GFP expression level. And then he was able to show the bright cells, which indicate high level of ID4 expression, transplant the best. That means in B, as you can show here, so because the cell not only have that ID4 GFP as a marker, but also have a ROSA LAC-Z with the beta galactosidase as another marker. So therefore, they can track the cells once they goes to the new testis, to the recipient testis. They can track the donor cells. And as here you can see that the ID4 EGFP bright cells give rise to more successful transplantation. But later on, scientists even find even more markers. For example, like a PEC7 and BMI1. Again, what is PEC7? PEC7 is a marker for satellite cell, undifferentiated satellite cell. You see, these markers are shared between adult stem cell types. The PEC7 is a marker for the transcription factor for the ultimate SSC together with BMI1. So basically, let me read this sentence so I hope you will understand that. AS cell, a single cells expressing either PEC7 or BMI1 also co-express ID4 will be the ultimate SSC. So for example, like when you have a PEC7 plus and ID4 plus cell, that will be the rarest among the AS spermatogonia subtypes. That's 0.25 of the total testicular cells. So that are considered the ultimate SSC. So you have three proteins that can mark the SSC, but here it's a strange combination. They should express either PEC7 or BMI1 and together at the same time together with ID4. That's the markers used to describe the ultimate SSC. Finally, in the last slide, I'm going to give you contradictory opinions or experiments related to whether there is female germ stem cells. But first of all the female follicle cells, they're from the primordial germ cell. I repeat that. So they're originally from primordial germ cell. But it's debatable whether there is a stem cell population in the ovary. I guess, if you consider the situation, you wouldn't think it is necessary to have a stem cell in the ovary. The reason is because it is long known that a woman at born will have 1 million of follicles. So basically, they're going to have 1 million of follicles at birth, but they will only ovulate about 400 times in their lifetime. Okay, so let's say every time one oocyte is released by ovulation, or maybe at most two. So in rare situation, you can have multiple, but it's very, very rare. So you have a million reserve as the reserve, but only 400 eventually gets released. So what's the point to even have stem cell, right? Because you already have a million follicles in there that can become oocyte, but you only use 400 or so. Therefore, there is no point to have a stem cell in there. Not like in males. In the testis, basically, they have to have a lot based on the amount that it gets released. But here, it doesn't seem necessary. But this scientist, Jonathan Tilley, in 2004, have an important publication. Basically, what his group is doing is they're trying to study follicular atresia. That's the process involved for the loss of the majority of follicles throughout life. That's called atresia. So because you have a million follicles, but they get lost throughout life gradually. So that process is called atresia. They studied atresia, but they found by labeling those follicles, they found that the process of losing the follicles are much more rapid than we have appreciated. Therefore, they feel like if you don't have a stem cells, the follicles will get lost very, very rapidly. So not 30 years, 35 years, like for most women. It will take a few years and then all the follicles are gone. Therefore, there must be a stem cell population there to replace the lost follicles. That's their opinion. Okay, and then they showed some evidence. And then they even showed that they transplanted fertile cells toward infertile animals to show, okay, now if you can rescue that infertility, that means there is a stem cell population in there. Of course, they didn't transplant the follicle; they transplant other cells in the ovary. They described that those other cells are the stem cell population. Therefore, after transplantation, you can make the infertile animal to become fertile again. But I would say that the majority of the scientists feel that the paper is misinterpreted. It's wrong. There shouldn't be female germ stem cells. And here I wouldn't bother to go through what they described in their paper. This is the key figure of the paper by Jonathan Tilley. But I guess eventually, if we have to demonstrate the presence of female germ cells, stem cells, ask that question I told you to ask. Can you demonstrate that a single cell that you isolated, is not a mature follicle, right? It's a single cell outside of a follicle or somewhere within the follicle, but people don't describe those as oocyte, immature oocyte. You isolate that cell, and then by genetic tracing, can you demonstrate that give a colony in another animal, and make that animal, if it is infertile, to become fertile again? So the single cell origin is very important in answering this type of controversial theories. Okay, that's the end of today's lecture. And if you have questions, you can stay behind and ask me.
Hey, Professor, I do have a quick question. It's about the group project, actually. So I'm from group 3, and I think we present next Friday, and I was wondering when we're going to go ahead and get our research article.
What?
So
I already post on the Sundays or Monday. So I think if you are group 3, you can expect to have that this Sunday or next Monday.
Okay, I gotcha. All right.
Okay.
Thank you. Sounds good.
Thank you, Dr. Lee, for the lecture. I think it was pretty straightforward.
All right.
Thank you so much.
You're welcome.